Exercise for the chapter biomolecule, and today we'll be doing question number 10.11 to 10.15. So first question, what are essential and non-essential amino acids, and give two examples of each type. Now we know that amino acids are the building blocks for polymer protein, and these amino acids are of two types depending upon whether they are synthesized within the body of an organism or not. So those amino acids which cannot be synthesized in the body and therefore they must be obtained through diet are called as essential amino acids. And the example of essential amino acid is valine and leucine. Okay. Now those amino acids which can be synthesized in our body and therefore they are not supposed to be supplemented with diet, they are called as non-essential amino acids. And two examples of these are glycine and alanine. Okay, so this is all about essential and non-essential amino acids. Now next question, define the following as related to protein. Okay, first one is your peptide linkage or peptide bond. So what is peptide linkage? We know that as discussed in the last question, that proteins are made up of amino acids, and these amino acids are acting as a monomer which combine to give you polymer protein. So these amino acids are connected to each other via bond which we call as peptide linkage or peptide bond. So peptide linkage or peptide bond connect different alpha amino acid to form polymer called protein. And if we see what is chemically, then chemically a peptide bond is amide linkage formed between carboxylic group of one alpha amino acid and NH2 group of other by the loss of a molecule of water. So this is how we are having peptide linkage formation. This is a molecule of glycine and here we have the molecule of alanine. We can have a peptide linkage between same type of amino acid. Here we have taken example of two different, and this is the carboxylic acid group of glycine and this is NH2 group of amino group of alanine. So we will be eliminating a molecule of water, that is your H2O, and whatever residue is left, it will combine to give you a peptide linkage. So it is basically an amide linkage, a link or the bond which is present in case of amides, and this is called as peptide linkage. Now here two amino acids are connected by a single bond, so it is called as dipeptide. Why we call it as dipeptide? Because two amino acids are involved in this case, and this dipeptide is named on the basis of amino acids which are connected together, so it is called as glycylalanine, and this is three-letter symbol for the dipeptide. So this is all about peptide linkage. Second one, second is primary structure. This is again in relation to protein. Now proteins, they have specific activity or specific role in the body of any organism, and that role depends upon its structure or its 3D form. So the very first elementary structure is the primary structure. What is primary structure? When different amino acids are connected by your amide linkage, then the polymer which is formed is called as the primary structure of the protein. As here we have given example of a dipeptide, and here this is an example of a polypeptide when n number of amino acids are connected by amide linkage. And how we have amide linkage in this case, you can see here, this is your connection between two group. This is first group. This is amide linkage here. The similar type of linkage here we have on this side also. So primary structure is how we define it: the specific sequence in which various alpha amino acids present in a protein are linked to one another is called as its primary structure. And one thing very important to remember is that any activity or function of the protein is because of its structure. So any change in the sequence of amino acid create a different type of protein. Okay. This is all about primary structure. Now, next is the third one. It is denaturation. Okay, denaturation you must have experienced in your daily life. Like, for example, when we boil egg, then egg white, which is transparent gelatinous, it will become opaque, and this is called as coagulation of egg white. So basically egg white is nothing but mainly protein, and when we heat, the heat brings about changes in the structure of protein, and therefore we get your thick opaque mass in the form of hard boiled egg or egg white. So why this happens? So when any protein in its native form means every protein has got a particular 3D structure, and according to that 3D structure it is having a particular role. Okay, so this form, any form 3D form of protein is called as its native form, where it has got some biological activity. So when a protein in its native form is subjected to physical change, like change in temperature. So if we heat anything, like here we are heating egg white or egg, or chemical change, for example, like change in pH. Okay, so the hydrogen bonds are disturbed. Your proteins, they have different structure. Proteins, they have got the primary structure, secondary structure, tertiary structure, and secondary and tertiary structures are basically because of the hydrogen bonding. So when we are heating or when we are treating with any chemical, like there is a change in pH, what happens that their secondary or tertiary structure get disrupted because of what? Because of the breakdown of the hydrogen bond on heating. Okay, so due to this, globules, which is tertiary structure of a protein, they will unfold, and a helix, which is secondary structure, that will get uncoiled, and a protein loses its biological activity. This is called as denaturation of protein. Okay, so examples are two given here. One is coagulation of egg white on boiling, and curdling of milk because of the presence of lactobacillus bacteria in the milk. And in this case, second case, there is a change in pH. We have acidic medium. So this is all about denaturation of protein. And if with diagram, if we want to understand that how denaturation takes place, so if we take example of any globular protein is just like that. We have such type of globule. So when for example, in case of egg white, what happens when we are heating? After heating, we will be getting the strands, or the globule will open up. In case of the helical structure, like we are having one polypeptide chain which is winded with another one. So again, in this case, what we have in case of denaturation, then these two chains, they will get separated. Okay, so this change is because of the change in either temperature, or for example here we are doing change in pH. So this is all about denaturation. Come to the next question. What are the common types of secondary structure of protein? So we know proteins have four different type of structure, mainly primary, secondary and tertiary. Sometimes quaternary structure, fourth degree structure is also there in case of some specific protein. So what is secondary structure? The secondary structure of protein refers to the shape or conformation in which a long polypeptide chain can exist. Okay, so different amino acids, they join with each other. Like for example, if we show this arrangement like this, so for example, we have one amino acid. This is one amino acid. Then we have another amino acid connected with each other. Then we have the third amino acid connected with each other, and so on. We will continue the polypeptide chain. So we are having different type of amino acid. This is polypeptide chain. Okay, now this polypeptide chain which we shown here, this will undergo some changes, and these changes are because of the hydrogen bonding. And after hydrogen bonding, we are getting two type of structure. One is alpha helix, another one is beta pleated sheet structure. Okay, so these two structures are what? They are the secondary structures of protein. And why these structures are formed? Because of the hydrogen bonding. So how we have hydrogen bonding in these two? First diagram is of alpha helix. So there we have two polypeptide chain which are wind around each other. Have a look here. We are having this oxo group of one amino acid, and this is NH group of another one, and in between these two we are having this hydrogen bond. Okay, so this is what you call hydrogen bond, or here we are having hydrogen bonding. Same we have in this case, or we are having here also hydrogen bonding. So because of this hydrogen bonding, we are having this secondary structure we call as alpha helix. Similar type of hydrogen bonding is present in case of the second structure, which is sheet like structure. And here, have a look here, this is the hydrogen bond, this is hydrogen bond, and this is hydrogen bond. So these structures, that is secondary structures, they exist because of what? Hydrogen bonding between different amino acids, and we get secondary structure of the protein. Next question. What type of bonding help in stabilizing the alpha helix structure? We have already done in the last question. So the alpha helix structure of protein is stabilized by intramolecular hydrogen bonding. Intramolecular means within the molecule. Okay, intramolecular means within the molecule. And what we have this hydrogen bonding between oxo group of one amino acid and NH group of another amino acid, and the number which we have, the which amino acids are connected, like if first amino acid is connected with the fourth amino acid residue in the chain via intramolecular hydrogen bonding. So this results in either coiling or connection between different polypeptide chain to give you secondary structure of protein, that we call as alpha helix structure of protein. Next is differentiate between globular and your fibrous protein. These are two types of protein. Like for example, again, these are tertiary structure of protein. So what is globular protein? As the name suggests, globular protein means the protein is resemble the shape of a sphere, or it has got spherical shape, while the fibrous protein, they have thread-like structure, as the name suggests. And your globular protein, they are soluble in water, while the other one, that is fibrous one, they are insoluble in water. The globular one, they possess biological activity, and therefore they are, for example, they exist in our body as enzyme, for example, invertase, your hormone, like insulin, or another very important globular protein, which is hemoglobin. Hemoglobin is the one which carry oxygen. So all these are the examples of globular protein. Now, next is fibrous protein. So, if we compare the biological activity, then fibrous protein, they do not have any biological activity, but they mainly exist as a structural material. Okay. And examples are the keratin in skin, and your hairs, your nail, collagen in tendon, and myosin in muscle. These are all structural materials, so they are made up of fibrous protein. This is all for today. We'll continue this chapter in the next video.